So hello friends, today we are going to discuss the second lecture on the which is based on the protein. Protein. So since in the first lecture I have discussed the its definition and regarding the peptide bond, amino acids and type of amino acid. So in this lecture we will discuss the uh, lecture first. After that we will start the remaining part. So if you see here, it is protein. The term protein is given by Bergelius. Molecular studies done by molecular studies is done by Mulder. And both scientists is awarded by Nobel Prize. Both scientists is awarded by Nobel Prize. So it is the great discovery regarding the proteins. And contribution is given much more towards the Bergelius because they coined the term proteins. So and the point is the protein is the polymers of amino acids. Polymer of amino acid. Polymer of amino acid. So you should be know about the what is amino acid. So amino means amine part, amine is NH2, acid means COH part. Another group is G, which may be variable, and hydrogen. This is alpha hydrogen. So here I have attached G. G may be either aliphatic, aromatic and heterocycles. So on the basis of G, amino acid is further classified. Here either aliphatic or aromatic heterocycles. So you should be know also aliphatic means either carbon hydrogen will be present, not either in this. It either a straight line or cyclic. Straight line means this one and cyclic means this one. So maybe like that it is called aliphatic. Aromatic means there will be conjugation. Like that benzene, it is the best example of aromatic. Heterocycle is in if heterocycle means it is the cyclic. The must condition is cyclic, and the another group besides carbon and hydrogen means there will be hydrogen is also present. So they may be either aliphatic or aromatic, like that. So it is the heterocycles. And the second condition is that in proteins peptide bond is present. Peptide bond, the chemical structure of peptide bond is CONH. So you should be know about the CONH. What is CONH? And how you can form the CONH. So if you see here, so first you should be know about the what is amino acid. So amino acid structure is this one. So if you see, this part comes from one amino acid. So I have attached NH. Suppose that this is the amino acid, so NH from this amino acid. So next is the carbon, this is the hydrogen, this is the G. And this is the COH. And this one CO. If you see the CO means one OH will be released out. So if you minus the one OH, and NS means one hydrogen will be released. H plus. So the remaining part will be CHG and NH2. So CHG and NH2. So means when water molecule is released out from between two amino acid, then this moiety will be form. Okay, so it is this is called peptide bond. Okay, so you can easily form peptide bond. Okay, so it is the basic points regarding the proteins. Okay, so it is the first lecture actually I have discussed before, so we will come on the second lecture. Okay, I think you will be understand very easy way. Okay, in second lecture in this lecture I will discuss the classification of amino, classification of proteins, classification of proteins. Okay, so actually protein is classified on the basis of polypeptide polypeptide chain, on the basis of polypeptide chain. Okay, you can also write down on the basis of on the basis of polypeptide chain, and second one is that on the basis of on the basis of structural structural protein. Okay, on the basis of polypeptide chain and the basis of structural protein. Okay, you can also see here on the basis of polypeptide proteins. Okay, so two classification here will be discussed. Okay, so first of all, if you see here on the basis of polypeptide proteins, on the basis of polypeptide chain. Okay, so on the basis of polypeptide chain you can classify protein is two types. First is monomeric proteins and second one is multimeric proteins. Okay, so multimeric, first of all you should be know what is monomeric. Mono means one, meric means one type. Okay, so if you see here, suppose that this is the monomeric proteins and in which one type of peptide bond is present. Here if you attached in clear way we will discuss this one. Okay, and this is the amino acids. Okay, this is the amino acid, this is the peptide bond, okay, so interaction will be, suppose that here CO is present. Okay, here NH is present. Okay, because in peptide CO NH is found. Okay, so interaction will be hydrogen, this one like that. Okay, so this type of interaction will be present. Okay, so it is the monomeric protein. Best example is lysozyme. What is lysozyme? First of all you should be know about that. Lysozyme is found first of all in tears and saliva. Okay, both are bactericidal means it kills the bacteria. Okay, it kills the growth of bacteria. Okay, so that's why it is called bactericidal. Okay, lysozyme, the second lysozyme. Okay, second is myoglobin. Okay, so globin, here if you see, globin is the protein part. Okay, myoglobin. Okay, so it is found in the muscles. Okay, so myoglobins. Okay, so it is also an example of monomeric protein. Okay, so I think you will be you can easily define that monomeric proteins and its example. Okay, the second point is that the second protein is, so we will erase this part. Okay, the second point is that the second protein is multimeric protein. Multimeric protein. Okay, so multimeric, what is the meaning of multimeric? Multi means several. Okay, meric means orientation, you can say. Okay, so if several types of amino acids, several type of peptide bond is present, many you can say many peptide bond is present. Okay, so many peptide bond is present, so that's why in this multimeric proteins many polypeptide bond is present. And best example is hemoglobin. Okay, if you see hemoglobin, four type of polypeptide chain, four types of polypeptide chain is present. Okay, so four type of, in this 2 alpha and 2 beta chain is present. Okay, so 4 2 alpha and 2 beta chain is present. Okay, second example is insulin. Okay, second example is insulin, means 1 alpha and 1 beta chain is present. Okay, so it is the best example of multimeric protein. Okay, actually in this I have also, in monomeric case, monomeric proteins, it is non-functional protein. It is non-functional proteins. Okay, it is non-functional protein, and these are the non-functional protein. But the exception in this protein is that exception is insulin. Insulin is monomeric protein but is functional. Okay, so actually it is that I have missed the important point, so I will discuss here, and so I have been discuss here. Okay, so in this protein, it is the functional protein. Multimeric protein is the, it is the functional protein. It is the functional protein. Okay, so it's the best example of multimeric proteins. Okay, so we will discuss the remaining another points. Okay, so it is all about the on the classification on the basis of polypeptides chain. Okay, the second classification is based on the structure. Classification based on the classification based on the structure of protein. Okay, so in this classification, it is second classification. In this, three type protein is categorized into four types. First is primary structure. Second is first is primary structure. Okay, and second is secondary structure. Third is tertiary structure. And fourth is quaternary structure. Okay, So it is the I will define what is primary, what is secondary, and what is tertiary, and remaining is quaternary proteins. Okay, so if you see here primary structure, we will discuss the first primary structure. Okay, it is first point. So primary structure means, in this, suppose that this is the amino acid joined in a linear way. Okay, in a linear way means 180 angle. Okay, so it is called, you will understand that it is amino acid, and this one is peptide bond. Okay, peptide bond. Okay, so it is the linear structure, and it is non-functional non-functional proteins. Okay, non-functional, but exception is here insulin. Okay, it is functional. Okay, it is functional. Okay, so it is the some basic points of primary structure of amino acid, primary structure of protein. Okay, so we will discuss the secondary structure. Okay, in secondary structure, suppose that secondary structure, two polypeptide chain is present. Okay, two polypeptide chain is present. Okay, like that. Okay, so if you say this is the chain of proteins. Okay, this one is alpha, suppose that this one is beta. Okay, so first of all, it is helical type, the structure is helical. Okay, you can say helicity of this. Okay, so it is the helical structure, and suppose that this one. Okay, this one, what happened here, this is the amino acid, this is the OH. Okay, means here will be, you can see in this protein hydrogen bonding is present. Suppose that one amino acid containing OH, another NH. Okay, so interaction will be on this one. Okay, so these are the amino acids. These are the amino acids. Okay, so if you cut the sum, I will cut this part. Suppose that I will cut this part. Okay, so these are the amino acid, this is the amino acid. So suppose that this one. Okay, so I will draw the structure of amino acid NH. Okay, CH, and remaining part is G. Okay, G, and it is the NH. Okay, suppose that this. Okay, this is another NH. Suppose this is another CO NH. Okay, NaCH, G. Okay, and CO part. Okay, so like that, it is helical type, means this structure you can chemically can see. Okay, so in this, what happen, interaction will be interaction, this type of interaction actually is called hydrogen bonding. So interaction like that. Okay, oxygen will interact in this, oxygen will interact this way, nitrogen interacts another below it. Okay, so it is called hydrogen bonding. Okay, so in this the main point is that, first of all, polypeptides present. Polypeptide, it is a type of polypeptide chain. Okay, and hydrogen bonding is also present. Hydrogen bonding is present. Okay, so it is the, and it is helical type of protein. Okay, okay, so best example of this protein is example is keratin. Okay, keratin is the best example of secondary structure of proteins. Okay, so it is the all about. Okay, so it may be alpha helix, you can say it is the alpha helix model. Okay, okay, so in the second, in the remaining part is tertiary structure. The remaining part is tertiary structure of protein. Okay, so it is the third type of structure. Okay, so tertiary, if you see here, tertiary means, suppose that this is the polypeptide chain, this is also polypeptide chain, this is also protein structure. Okay, so in this, if you see here, suppose that this the OS part remaining, and in this NH2 part, like that. Okay, this is NH2, this is OH, this is OH, this is NH2. Okay, so these, so, okay, if you see in the biological system, receptor is also found. Receptor, okay, receptor means in the in bodies there's lots of receptor is present, and these present like pockets. Okay, in this, these type of chain is present like that. Okay, and in this part, these OH and NH2 group will be outgrowth is present. Okay, like that. Okay, so any drug molecule when comes near about, they will interact from this. Okay, okay, so one drug, suppose that this is the any drug, suppose that where H is present, where OCS3 is present. Okay, so it will interact from these pockets. Okay, so it is called functional protein. Okay, it is called functional protein. Functional protein. Okay, and in this hydrogen bonding is present. Hydrogen bonding present. Okay, so it is the main point regarding this protein. Okay, and okay, so this site is also called active site. Active site. Okay, so it is space, each and every. Best example, if you see here, example is enzyme. Okay, so it is the, in this active site is present, it is functional protein. In this hydrogen bonding is present. Okay, in receptor, in enzymes, enzyme receptor is proteinaceous nature, and it is the name is enzyme. Enzyme is proteinaceous nature. So these proteins will be present below the receptor, and outgrowth of functional moiety will outgrowth it outside. Okay, so that's why any drug molecule will attach from this part. Okay, it is 3D drug. Most of the drug is three-dimensional. Okay, three-dimensional means, suppose that this it is below, suppose that any group is below G1 and one is above. Okay, means this is the board, it is 2D. Okay, and above one group if you attach from this board, and below one group is present, so this is called 3D. Okay, so this type of, these are the such type of proteins. Okay, so it is very important proteins. Okay, the remaining is tertiary structure of protein. Tertiary or quaternary proteins. Okay, quaternary protein. Okay, quaternary protein, if you see, best example is hemoglobin. Hemoglobin is the best example of quaternary proteins, and there is a similar like the tertiary proteins, like the one polypeptide like that. Okay, so it is called quaternary proteins. Characteristics is similar to it is specifically functional. Peptide bond is present. Peptide bond is present. Disulfide bond is also present. Dis in between. Disulfide is also present. Disulfide bond is present. Okay, so it is the main point is that disulfide bond is present, and it is similar to tertiary protein but is specifically functional. Okay, best example is hemoglobin. Okay, so it is the classification I have discussed. Okay, so classification here end up. Okay, so I have erased it. Okay, 2 point is also remaining regarding the protein. The protein is two types actually on the basis of first class protein. Some protein actually two type of protein is present. First type of protein and second type of protein. Okay, in first type of protein, both amino acid essential as well as essential and non-essential amino acid is present. But in case say in second type of protein only, either there will be deficiency of either essential or non-essential amino acid. Okay, so it is the two type of protein in actually in competitive exams. In one time I think in PMT one question is asked that what is first type of protein. Okay, so you can understand that in first type of protein both uh, essential and non-essential amino acid is present, but in second case only either deficiency of essential or non-essential will be. So first type of the best example of first type of protein is animal proteins. All in animal proteins both essential as well as non-essential amino acid present, and second type of protein is example is vegetables. Okay, so if you eat vegetables, then either deficiency of essential or non-essential amino acid will be present. Okay, so it is so overall my lecture of regarding the protein is end of here, and please watch before this lecture. First of all, you should be watched regarding the proteins, amino acids and type of amino acids. Okay, then classification, then you will be understand in a better way. Okay, so thank you friends, and please like and subscribe my channel. Okay.